Welcome back, everyone. In this episode, we're going to look at gustation right after olfaction. First of all, we're going to see an overview of gustation, then talk about the transduction of gustatory information in the taste buds. After that, we will discuss the encoding of gustatory information, and then go to brainstem nuclei and the gustatory system that is located in the cerebrum. This is the map for today. We go from taste cells through 3 cranial nerves to the solitary nucleus in the brainstem, and then to the thalamus and the cortex. Here is an overview of gustation. Like olfaction, gustation is a chemical sense. Its stimulus is chemical particles. For the salty taste, we taste sodium ions. For sourness, we taste hydrogen ions. For bitterness, there are various molecules that trigger this sensation. Usually, these molecules are toxic for us, bad for our health, so that's why we sense it as bitter so that we don't eat it. Sweetness is triggered by sugars and some other molecules, while umami is usually triggered by amino acids, usually glutamate. For example, monosodium glutamate is an additive that we usually use to make our food taste better. Saltiness, sourness, bitterness, sweetness, and umaminess are the 5 basic senses of gustation. We're quite sure about this. There may also be some other sub-senses of gustation. For example, we might be able to sense fat, starch, water, even carbon dioxide, such as those in soda. However, we're not so sure about these yet. Let's see where the taste buds are located. On our tongue, we have various papillae. These are the little budgets on the tongue. Contrary to what we may believe, the papillae are the little budgets on the tongue, and the taste buds are embedded within them. As we see in this diagram, the taste buds are located in the grooves of the papilla. This is to ensure that the fluids in our mouth can get into these grooves, so that the taste can linger around the taste buds for a longer period of time. The papilla may have different shapes: fungiform, foliate, valate, etc. The taste buds, as we have discussed, are inside these grooves. The taste buds are in these grooves around the papilla. They are mainly made of taste receptor cells and basal cells. There are also supporting cells. Taste receptor cells is the main cell type that helps with gustatory transduction. In the following sections, we will discuss how the 5 main tastes are transduced by the taste buds. Sourness and saltiness are transduced by ion channels, while bitterness, sweetness, and umami are transduced by G protein coupled receptors. So first, through ion channels. Saltiness, as we have said, is the sense of sodium ions. Here, the sodium ions directly flow into the taste receptor cell through these ion channels. Sometimes, a large anion in the salt may inhibit the salty taste. For example, if the sodium ion combines with an anion that tastes sweet, the sweet taste will dominate over the salty taste. Also, excessive sodium ion concentration may trigger sour or bitter sensations. For sourness, the situation is a little bit more complex. Instead of flowing right into the cell, hydrogen ions blocks potassium channels. This prevents potassium efflux. It also activates transient receptor potential channels, which allows the sodium ion influx. Both of these allow the depolarization of the taste cell. Please note that the taste receptor cells do not have action potentials. They only depolarize. However, this depolarization indeed triggers the release of neurotransmitters. Here, for saltiness and sourness, the taste receptor cells release serotonin. Now let's take a look at the G protein coupled receptors transduction of taste signals. This process is realized through dimers of GPCRs. For bitterness, T1R1 and T1R3 dimers are used. For sweetness, a dimer of T1R2 and T1R3 is used. And for umami, a family of T2R receptor channels are used. Let's take a look at the following steps in the transduction. For sweetness, a GS protein is used. As we have mentioned in a previous episode about synaptic transmission, the S in GS stands for stimulating. Here, the GS activates protein kinase A, which causes more cyclic AMP to be produced and inhibits potassium ion channels. This prevents the efflux of potassium ions and depolarizes the cell. For bitterness, a GQ G protein is used. It activates the phospholipase C, which converts PIP2 into IP3. IP3 then triggers calcium ion release from smooth endoplasmic reticulum, SER. The calcium, as we have mentioned in synaptic transmission, will bind to snare proteins and trigger the exocytosis of neurotransmitters. For umami, this process may involve various types of secondary messengers. In all three cases, there is a depolarization, a receptor potential of the cell. This triggers the release of neurotransmitters. Here, ATP is used as a neurotransmitter. Here we must comment that the spiciness of chilies is not a part of gustation. The spicy substance in many chilies is called capsaicin. Capsaicin is actually a nociception or temporal sensation. It is sensed by an ion channel called the TRPV1. As we have mentioned just now, TRP stands for transient receptor potential. This ion channel has very diverse functions. It can sense heat, injurious stimuli, and of course, capsaicin. That's why hot is actually a synonym of spicy. Similarly, the coolness of menthol in mint is not gustation. It is also a temperature sensation, and this is mediated by the TRPM8 channels, which is also a transient receptor potential receptor. This is the map up to this point. Saltiness and sourness are mediated through ion channels, while sweetness, bitterness, and umami are mediated, transduced, by GPCRs. These all occur in the taste cells. Now, it's question time on the transduction of gustatory signals. Please select the correct statements. A. Sweetness is not transduced via ion channels. B. In the transduction of sourness signals, hydrogen ions generally do not flow into the receptor cell through ion channels. C. Spiciness is sensed by the receptor TRPM8. D. Gustatory receptor cells that sense bitterness uses CAMP as a neurotransmitter. Please hit pause and think about this. The correct answers are A and B. Sweetness is indeed not transduced by ion channels. It is transduced by GPCRs. For B, the transduction of sourness signals does not involve hydrogen ion influx. However, hydrogen ions act on potassium and sodium channels and cause depolarization. C. Spiciness is sensed by the receptor TRPV1, not M8. TRPM8 is responsible for sensing the cool sensation of menthol. D. Gustatory receptor cells that sense bitterness uses ATP as a neurotransmitter. Usually, CAMP is used as a secondary messenger inside the cell. Going forward, let's discuss the encoding of information. The intensity, like olfaction, is usually encoded by the rate of firing of neurons. The identification involves a certain degree of population encoding. However, what is more important is that gustatory information is often combined with olfactory information. In fact, when we have a cold and our noses are stuffed, or when we pinch our nose and eat something, oftentimes we do not feel that the food is as delicious as before. This is because we are now lacking the olfactory information, which forms a very large part of the experience of tasting. Going forward, brainstem nuclei. To reach the brainstem nuclei, we have to go through several cranial nerves. There are 3 cranial nerves involved. The first one, the facial nerve, which is cranial nerve number 7. It innervates the anterior 2/3 of the tongue. The second one is the glossopharyngeal nerve, which is cranial nerve number 9. This one innervates the posterior 1/3 of the tongue. The third one is the vagus nerve, cranial nerve number 10. It governs the sensation of taste in the epiglottis and lower pharynx. Please note that these nerves have a lot of other functions, and tasting is not their simple only function. This is quite unlike the olfactory nerve or the optic nerve, for example, which only transduce the sensory information. Here, through the cranial nerves, we will reach a brainstem nuclei called the solitary nucleus. In the cerebrum, neurons from the solitary nucleus will travel along the solitary tract up to the thalamus. This process involves a degustation, which means that the axons from one side goes to the other. It reaches the ventral posteromedial, the VPM nucleus of the thalamus, which then sends information to the primary gustatory cortex G1 in the insula. The insula is a part of the cerebral cortex that lies inside the lateral fissure, which is the fissure that separates the frontal and parietal lobes and the temporal lobe. As shown here, if we use a clamp to pull apart the lobes, we can see the insula hidden within. The word insula means island in Latin, so it is like a little island inside the brain. In further processing the cortex, the information of gustation is integrated with olfactory information to generate the perception of tasting. This is the complete map of gustation, from taste cells to the solitary nucleus, then to the VPM nucleus in the thalamus, and finally to the gustatory cortex. The ultimate question time. Please answer the following questions. Number 1. Match the thalamic nuclei with the corresponding sensory modality. The thalamic nuclei are LGN, the lateral geniculate nucleus, VP, the ventral posterior nucleus, MGN, the medial geniculate nucleus, and VPM, the ventral posteromedial nucleus. The sensory modalities are gustation, vision, audition, and equilibrium sensation. Please match. And number 2, please name the cranial nerves that innervates the taste receptors in each of the areas 1, 2, and 3 as shown in the figure on the right. Here are the answers. LGN governs vision, VP is for equilibrium sensation, MGN is for audition, and VPM is for gustation. As for the cranial nerves, the area number 1 corresponds to the vagus nerve, cranial nerve number 10. Area 2 corresponds to the glossopharyngeal nerve, which is number 9. And the third area corresponds to the facial nerve, which is cranial nerve number 7. That's all for this episode. We have seen how gustation is, we have seen how the taste buds transduce gustatory information, how this information is encoded, and how the gustatory system goes into the brainstem and into the cerebrum. In the next episode, we're going to get to a very big sensory modality. In fact, it uses one of the largest sensory organs in our body. What will that be? Let's wait and see. Looking forward to seeing you in the next episode.